Warm welcome to this third webinar, which is one of the webinar series for HoliCare projects. But before we start, I would like to invite Dr. Mohammed to introduce our presenter today, and then we follow up with the presentation of today. Thank you. Over to you, Mohammed.
Yes, thank you very much. Thank you very much, Jean-Claude. I am Mohammed Bakait, so from now connecting from here, from Mast, Germany. And I have really all the honor to introduce my friend and colleague, Dr. Hamid Ismail, for this seminar. Very short introduction about him. So he's a graduate of the veterinary school where we went together. At that time, we graduated from the veterinary medicine, and then he took another path where he actually oriented himself towards the bioinformatics. He started then as he obtained then a master's degree and a PhD degree from North Carolina University, and worked some time as well also in Michigan Technological University. In the Department of Computer Science in a state-funded project. And right now he's back to North Carolina, and he's an assistant professor there in the university with the tasks of teaching undergraduate students bioinformatics and other courses like scalable computing and computational modeling and so forth. Those all are very big names. And yes, he also does research in the area of bioinformatics, in the application of machine learning in genomic data and so forth. So I'm very much sure that we would really benefit a lot from his knowledge today. To add more, Hamid, of course, is qualified and has a lot of experience as well in statistics, and he has also earned several professional certifications in different things like programming and clinical trials and so forth. He is also a publisher of very important reference books, 3 of them, in the field of bioinformatics, practical guides. And we have invited him today for this HoliCare Academy seminar to give us an introduction on the bioinformatic field, because it now turned to be very important to have a little bit of more information here within the activities of the HoliCare Academy. So thank you very much. And please, Hamid, you can start with this seminar.
Thank you so much, Dr. Mohammed, for your introduction. Then also I would like to extend my gratitude to organizers. This webinar is about introduction to bioinformatics. And the main objective is to give you introductions about bioinformatics and how to get bioinformatics data, and also how to and also we know something about the databases, and also we'll know the commonly used data format for in bioinformatics. And after that, also we'll discuss the some applications in bioinformatics like sequence alignment and applications of sequence alignment like value unique tree and plus. And so I hope this introduction to bioinformatics to be useful for those audience. And by the way, also I would like to thank audience for joining us in this webinar as well. So about bioinformatics, in fact, become one of the most important field in life sciences generally, because now has been used in medicines and in pharmacology for drug discovery, and also in biology as general for to interpret the for understanding some biological activities. And bioinformatics is the field that focuses on the analysis and the analysis of biological data, and also in development of algorithm, computation methodology to analyze this biological data. And the bioinformatics itself can combine the other field as well. Biology is the central field of science, but also there are other fields like computer science and mathematics, statistics, chemistry, physics, and any other fields that contribute to biological information and data analysis can be part of bioinformatics. So, of course, the central field is biology because we are getting these data from biology, and specifically we are focusing on the biomolecules. Biomolecules, that mean the DNA, RNA, and proteins, because those are the main or the primary molecules that we are depending on in bioinformatics. But also there are other parts, like from all these disciplines. Each one of these disciplines contributes in a specific way, like computer science, for example. They will develop algorithm, programming. They are using programming to analyze this data. And also they develop databases and hardware. So everything related to computers and computational resources and programming database. This actually can be done by computer science. Also in physics, the study of physicochemical properties of proteins, and in chemistry also as well. So all of them actually, they play together. And of course, we are using mathematics and statistics to analyze this data and interpret this biological data. So there is, in fact, very good bioinformaticians in general. Bioinformatics, we can divide bioinformatician to different level, but the good bioinformatician is supposed to be good biologist and computer scientist. And actually, this is not like, I don't want to scare those who, in a specific field, maybe biologist or computer scientist, but those things, they can be gained, or can be something that, you can do by yourself. So this is just, I'm going to give you a brief history about the molecular biology, or about the milestone in bioinformatics, or that made molecular biology and bioinformatics possible. And this is maybe the discovery of the structures of DNA, or deoxyribonucleic acid. This is one of the most important scientific achievement in the last decades. And when we mention the discovery of molecules of DNA, immediately we think about James Watson and Francis Crick, because those two scientists who discovered the structure of the DNA molecules. But in fact, there are some scientists before him a while ago, and the DNA first time actually discovered maybe in 1886 by Friedrich Miescher, who discovered actually the DNA when he tried to extract protein from white blood cells, and he discovered a new material, and he called it nuclein. And after that, other scientists also come to add to that knowledge, and one of them is the Russian scientist who could figure out the chemical compositions of the DNA, the nucleotides, and also he could discover that nucleotides consist of phosphate group and consist of the sugar which is deoxyribose, and base, a nitrogenous base, which is cytosine, adenine, guanine, and adenine, cytosine, and thymine. So these discoveries, and also there was another scientist who played a very important role actually to know, because when he was studying, when he extracting the material of DNA and tried to quantify the DNA, he found that the number of cytosine, the amount of cytosine is equal to guanine, and the amount of adenine is equal to thymine. So all these informations, after that, they tried to, James Watson and Francis Crick, they're trying to solve the structures of DNA using this information, but they were not able at that time, and they were using very complicated and very complex trying to calculation to try to figure out the structures of the DNA. And they couldn't do that only by the discovery of Rosalind Franklin's. She was a scientist working in X-ray diffraction lab, and she could capture the first image for DNA molecules. And that was in 1950. And James Watson and Francis Crick immediately, they benefit from that information, and they could figure out the structures of the DNA. Then that becomes the one of the most important achievements in scientific achievement in last decade. And from there, the molecular biology and all the advancements that we see actually depend on that discovery. So after the also there is another milestone. This Margaret Dio, this is the first bioinformaticians who developed a database for protein and for protein sequence and protein structures. And also she used protein sequence to study evolution. And she discovered the first substitution matrix, which is still we are using it now, which is the point accepted mutation, or PAM. You see, we are using actually. She was discovered by Margaret Diouf, and she was considered one of actually she's the first bioinformatician. And development is still was going on at that time. Then come the era of sequencing, and by sequencing, then they're able to find the sequence of gene molecules. Actually, that began with a Sanger method and Maxam Gilbert Sanger, and this is 1970. And after that, in 1983, the discovery of PCR, or polymerase chain reactions, still are using it to amplify a piece of DNA or gene, but also it's used in sequencing. So also it contributes to molecular biology, and this discovery now we are using it in many fields, in many technology. PCR one of them, but also is being used in sequencing and other applications as well. So another milestone is Human Genome Project, and this began in 1990 and ended in 2003. And they are trying to sequence human genome, and this project was international effort from different scientists from around the world. They are using different sequencing machine, Sanger method at that time, the only method that was they were using. And the project itself to sequence the complete human genome took like 10 years. And during this 10 years, like $3 billion of money were spent on that project. But the good thing about this, it's allowed scientists to be aware of the importance of genome, and the advancement of bioinformatics, molecular biology, new algorithms were developed, programmers were encouraged to develop programs and to be involved in bioinformatics. And the development of bioinformatics in fact, contributed to this, to Human Genome Project, because they are trying to understand Human Project. And after that, many technologies were discovered, especially the next generation sequencing technology like Illumina and other brand as well. And so high throughput sequencing like next generation sequencing and third generation sequencing, they are able to sequence a human genome or any genome in hours, and they reduce the amount from like 3 billion to like few like less than $1,000. And instead of like hundreds of scientists, a single scientist and single equipment can do that job. So this technology, massive data were accumulated, and this massive data actually help us to develop the bioinformatics, because bioinformatics, this data must be analyzed, must be interpreted, and that also new algorithms must be introduced to the field of bioinformatics, and computer science actually did very good job in doing this. So they are essential. They are very essential things bioinformatician must know, because we understand, especially till now, actually most of the bioinformatician from computer science, and they are some of them actually they don't have background in biology, but understanding molecular biology is very essential, because this is the part that we are getting information from. So the bioinformatics itself depend on the primary or basic things that bioinformatics depend on the three biomolecules: the DNA, deoxyribonucleic acid, and RNA, nucleic acid, and proteins. So those are we are getting information from this, because we are getting we are beginning bioinformatics by studying the sequence of these three biomolecules. And about DNA specifically, it consists of nucleotides, and also we know about the base pair, like cytosine bind to guanine by 3 hydrogen atom, hydrogen bond, and adenine with thymine. So there is a DNA consists of 2 strands, and there is a complementary rule for that. Okay, and these 3 nucleotides consist of phosphate group, which consists the backbone, and sugar, deoxyribose, and bases. The bases, or nitrogenous bases, this which distinguish the 4 nucleotides. So we forget about the other part of nucleotides, and we focus only on the bases, which are thymine, cytosine, adenine, guanine, and we consider this as alphabet of DNA. So when we're talking about the DNA sequence, that means our focus will be on this, because this will be the alphabet, or the alphabet of the sequence of DNA. The same thing here also for RNA. The alphabet, the only difference is the RNA is single-stranded. It's not double-stranded, it's single-stranded, but also it can form some kind of structures, structure, like structure like ribosomes or transfer RNA, or it can take some structure to perform a specific function. Okay. And of course, we have messenger RNA, and messenger RNA, this is a protein coding, and the most study depend on messenger or mRNA. But the messenger RNA as general, the only difference in nucleotide is uracil. Okay. And so we don't have thymine, we don't have T, we have uracil. So the alphabet of the RNA is A, C, G, or U. Okay, so this when we sequence. And what also I want to add here, because if you are working in a biotechnology lab or molecular biology lab and you extract RNA, so immediately, in most cases, you will convert your RNA into cDNA, or we call it complementary DNA. So when you are working in bioinformatics, you may not find uracil, but instead you will find T. And the reason, because you must convert it into complementary DNA, because the RNA is not stable. It's very easy target to some enzyme like RNase. High temperature and some chemical will degrade it. So once we extracted the RNA in the lab, immediately we convert it into complementary DNA using the process, what we call it transcription, reverse transcription, or enzyme reverse transcriptase. So, and the third molecule is proteins, and proteins consist of the 20 essential amino acids. And again, the alphabet is, we are giving each of these amino acids, we give them a one letter. As that single letter will be the alphabet of this. So when we are dealing with the sequence of protein, we are dealing actually with this letter, and each one represent a protein. And protein itself, it has, actually it has like a secondary structure. They have different structure, linear structures. This is the normal sequence, but also it can form 3D structures. Then 3D structure can be secondary structures, which consist of alpha helix, beta sheet, and also it can be tertiary structures and quaternary structures. And the idea, because the protein itself, it must be converted into 3D dimension to do its job, its function, because this is the fun. So this is the transformations of genetic material from the DNA to protein, and protein, the one who which does its function. So in the classic classifications of living organism, and there are three domains: bacteria, archaea, and eukarya, and that is fine, actually, that classic. But when you study genomics, and genomics, because we are getting information from genomic data, so for us here, the organism, the cell itself, the source of those three biomolecules we classify them into prokaryotes and eukaryotes. So in the prokaryotes include bacteria and archaea, and eukaryotes include protista, fungi, plants, and animals. And the division here because of the nuclear material. Okay. In case of prokaryotes, they don't have definite nucleus, and we find the genome, and the genome, the complete set of DNA molecules is the cells. We find it floating on the cytoplasm. But in case of eukaryote, we will find a nucleus with a nuclear membrane, and inside this nucleus, we will find different chromosomes, the DNA. But we were talking about the genome, and this is the molecules of DNA in the cells, and of course, like eukaryotes, this is multicellular organism. All cells, they have the same set of DNA. All cells have the same set of DNA, and all cells, they have, except in case of animals, red blood cells, they don't have a nucleus, so to do its function. So for prokaryotes, the genome of prokaryotes consists of its lack of a nuclear membrane, but the genome consists of the DNA that's floating on the cytoplasm. In addition to, in some bacteria, you may find a circular DNA we call plasmid, and that plasmid also is very important and especially play in resistance of bacteria. There's some genes there, and maybe we'll discuss this. And in case of eukaryote, the genome consists of the nuclear genome and mitochondrial genome. This is in case of animals. But in case of plant, instead of mitochondria, we'll find chloroplast. So these information must be known for bioinformatician, because when study genome or dealing with sequence, they should know what the nature of this DNA. And for virus actually is not a living organism, but it is a particle. It's not a living organism because he has no protein production machinery. So it must invade and use the host machinery to produce protein, whether it can be RNA or DNA. This is the genome, and when invade the cell, it may be integrated to the genome of the host, and after that it will express its genes and to produce proteins and so on. So these three different entity, prokaryote cells and eukaryote cells and virus particles, those are the primary sources of our bioinformatic data, which are, sequence and so on. So now, after we know the importance of these three molecules, so what is the link between those three molecules? In the genome, we say the genome is all the set of molecules, chromosomes, any pieces of DNA inside the cells is part of the genome. But only a small portion of the genome is functional. Others are not functional, but of course they are functional, but maybe their function is not discovered yet. So you cannot say they are not functional, okay? But by functioning, we mean the portion of protein that can code for protein or has a specific function. So in case of human genome, we have maybe like almost like 97%. We don't know about it. Before they used to call like junk DNA, we are not calling it anymore. But 3%, those divided into they consist of functional units. We call them genes. So these genes, in fact, code for messenger RNA. And all this, we call them the central dogma of molecular biology. That means the DNA can be transferred into RNA, and RNA translated into protein. And there are some, so this is unidirectional, but there are some exceptions. Sometimes RNA also can be transferred to DNA, and the process is, we call it reverse transcription, because there are some can be converted into DNA and integrated in the genome again, and that actually lead to elongations of DNA. And now, if we look to the human DNA, we will find many sequences before they were RNA, but they are inserted there. And so the movement here, the genome, there are about 3% of them, they are functional, and those function produce RNA. And there are maybe 4 types of RNA. One of them, messenger RNA, which is protein coding, and the second one is ribosomal RNA, which form the ribosomes, and transfer RNA, which bind to amino acids and help in the translation, and microRNA, which play a very important role in gene regulation. And so messenger RNA is specifically, so this is the only protein coding DNA. This translated because they form what we call it a reading frame, and each 3 nucleotides of this reading frame, we call them codon, and each of these codon can be translated into amino acids. Okay, there are redundancies in this, but at the end, they are using these genetic codes to translate into amino acids, and then the amino acids, of course, this translation will take place in ribosomes, and protein will be formed, and after that, the protein will take its 3-dimension. Protein cannot function without 3-dimension, and after that, it will be mobilized to the compartment inside the cell, or maybe extracellular to do its function. So what is the importance here? We can link always between what we call it genotypes and phenotypes, because in genetic material, there are some kind of the gene are there, and in the gene, there might be some kind of mutation like SNP, single nucleotide polymorphism, maybe indel, insertion or deletion, or maybe copy number variants, and all these contribute to the diversity of things and between individuals within the species, and that lead to phenotypes. So always there are linked between genotype and phenotype that can be like shape or trait or maybe disease or condition or anything like that. Okay, so this is the link between that. It is very important to know the difference between eukaryote gene and prokaryote gene and the gene structure itself. In case of eukaryotes, eukaryote gene, there is intron and exon. Exon, this is the only, this is the protein coding part. Intron is not protein coding part. In case of prokaryote, there is no intron. Okay, there is no intron. In case of eukaryote gene, each gene will be controlled by a single regulatory region. Okay, so it is have a region that can regulate the gene expression. But in case of prokaryotes, you will find a group of genes. All of them, they control together. And all of them in a container, we call them opera. Okay, so this is the difference, and this is very important difference when you come to analyze. Always think about what kind of disease is it, eukaryote or eukaryote or prokaryote. Again, the structures of eukaryote gene, because the intron, this lead to very important thing actually, because a single gene can be reduced multiple protein, okay, due to alternative splicing, okay, because you know this intron, sometimes the exon can be rearranged, or part of the exon can be included to be part of intron can be included to be part of exon and part of exon, and they can create a different reading frame, and this reading frame will produce completely different protein. We call them isoform of protein, and this is something that also we should know. So there is really very important thing that's behind the diversity, mutation, evolution, everything that now dealing with is DNA mutation. And mutation is always can consider be as random because there is some kind of errors that can take place. And also it can be due to mutagen. Mutagen can be chemicals or can be radiation or anything like that. But mutation is all the time, is there, and mutation lead to generation to diversity within the species. It can take to new species in evolutions, and also it can lead, to different phenomena. So what the mutation is, if the sequence of the DNA change by error or by mutagen, then we call that mutation, and mutation can be base substitution or nucleotide substitution, or nucleotide substitution. That means a nucleotide or base will be replaced by another base. And later on, we will see if there is any consequences, because when the mutation can take place in different parts of the genome, can be, but the one with consequences in the protein producing sequences, because that means they are going to change the sequence of protein, and also they may have some implication, in the health or trait or something like that. So substitution, we have substitution, deletion, when a sequence or base deleted, insertion if a nucleotide inserted, and there are the consequences are different. Because if mutation, because we have some kind of redundancy that a codon, different codon may code for a single amino acid, but sometimes mutation maybe doesn't cause any kind of change, so it still will get the same amino acids. And in this case, we call it silence. But if the amino acid change to different one, then we call it missense. And the consequences may be different, because it might take to disease, maybe it takes to favorite trait, or maybe bad trait, but that depends on the location, what exactly the effect of that. And again, the most important on kind of that called what we call them frame shift. And frame shift can be by delete because it frame because the transcript divided into codon. So if we miss any one of bases or nucleotides, that means they will change the protein. So we'll get a protein, we don't know exactly its function. It might be good, it might be bad, but most of the time it might lead to some consequences like disease. Most of the cancer come actually through this frame shift. Nonsense, when the mutation lead to stop codon. Once it leads to stop codon, then will lead to that will lead to truncated protein. Protein will not be complete, it will be functional. So, and finally, we find like translocations, and translocation piece of chromosomes. They can exchange the pieces, and some of them actually they are not lethal, but some of them actually they have pathogenic effect that depend on if there are any genes affected. So mutation actually lead to diversity and evolution. And now we see within these species, whether like humans or cats or dogs, we find different diversity. And diversity in colors and shapes and different, and all this because of mutation, because of the SNPs, because of some kind of mutation that lead to this kind of and evolution itself, as now actually it's become like almost like a fact that those living organisms are actually diverging from its common ancestors. And because of mutations and because of speciation, lead to a different, different species. So this diversity of these species actually due to evolutions and evolution due to mutations. And of course, there is a factor of natural selection. And natural selection that if mutation is good, and so that will impact the reproduction and survival of organism. That means it will survive, and if the mutation is bad, that the natural selection will lead to eliminate that trait and also that organism. So this just a general. But generally, we will go to what we call it homology and homolog. That means most of the things, they diverged from a common ancestors. And when they diverged from common ancestor, always you have to think about the two things. Because it's all about genes, right? Because genes which lead to this phenotype, lead to genotype, lead to change of biological activities, shape, everything actually controlled by this kind of gene. So gene duplications take place in the same organism. And after that, maybe after a long time, like globin, for example, this early globin gene was supposed to be this is the ancestor gene of the alpha, beta globin, and like hemoglobin and myoglobin and cytoglobin and different globin. They think actually this is the origin of, this is the common ancestor of this long time ago. But because of duplication, they diverge into, for example, like alpha chain gene and alpha chain gene. And after that, this for when we speciation, that means each species develop. So each of these species will have its own gene. And so we call this, so generally homology, homologs can be divided into ortholog if they are in different, if that there are similarity between genes in the same species, and paralog if they are in the same species. So similarity will be in the same species, and this happen by gene duplication, while the ortholog by speciation. And homology can be paralog and so on. So this some information. So now we'll talk about bioinformatic data acquisition, and this part of really is very important because biologists, researchers are general. Sometimes they think focus how we get bioinformatics data. Okay, so this question is valid, especially for newbies, for these new scientists. They need this bioinformatics data. So bioinformatics data, we can get it either from laboratory, from the lab experiment. We can design our own lab and research, and of course that we did for the experimental design that depend on what exactly we are studying. Okay, well, if maybe we want to study a specific gene, then we call that target gene sequence. We can do target gene sequence, whether to use, just normal PCR polymerase chain reaction, or we can use high throughput sequencing like next generation sequencing. But the idea here, our focus will be on a specific gene or genes. Okay, we target them, and we can think also we can plan for all genome sequencing and all transcriptome sequencing. Transcriptome sequencing that we need to extract the RNA in the cell, all RNA, or maybe metagenomics, if we need to study environmental samples or clinical samples that include many microbes. We need to identify the microbes in these samples. And they have many applications for this in medicals, in ecology, in to study environment, and to understand the ecosystem. Even the ecosystem, millions of years ago, is still that using this kind of techniques to understand exactly what organism at that time. Epigenetics, if we need to study something, like transcription factors and modifications of histone. Those kind of question that come up while studying in the lab, and then we will collect our samples, of course, from a biological, biological entities, and we extract our DNA or RNA or protein, and after that we will generate this data by using sequencer. And sequencer, some labs they have sequencer, and some they send to other company to sequence for them. These many scientists, actually, most of the scientists, especially in the wet lab, they are doing this. And after that, when they get back the data, immediately they will think about bioinformatics, because now they acquire the bioinformatic data sequence. But after that, what to do with it? It will be bioinformatics, right? So now we're getting from lab, we get this information. So, but sometimes maybe you don't need to go that path, and because already we have during the last decades millions of sequence and biological data already deposited in database, and maybe you can find you can depend on, instead of lab, if that also depend on what exactly kind of scientific questions that we need to answer, maybe you will find that data in a bioinformatic databases. And this in case you can get from a databases, you will find all kinds of sequence, the raw sequence, that sequence we are getting from a next generation sequencer, or third generation sequencer, or Sanger sequencer, or microarray, or also you can find other information like primary, secondary information, like the relationship between phenotype and genotype and phenotypes and structures of proteins. And you can find also information about diseases like online Mendelian inheritance for men and so on. So you will find, so when you need to think about the data, if you are not working in a wet lab, still you have option. Now, actually, we have found millions and millions of COVID-19 sequence deposited in a database we call the GISE. We have millions and millions for any part of in bioscience, life science. You will find a database that collect, store, and that information are ready for you, or it just you need just to and also those databases are provided with a way to search to find the right data that can serve your research. So this and when we talk about the next generation sequencing, then this one of the main sources. So this one of the main sources. Like always, we know we sequencing, because when we get the samples, of course, we need to fragment it and we add adapters, and after that we bind it. But the idea of next generation sequencing is that we will get very massive raw data, and that raw data will get it in the form of FASTA files. Then next generation sequencing actually is become the main source for this biological data. And in fact, there are different. We can get data from, especially from next generation sequencing. We can get it from a different, like whole genome sequencer, and we can get like variant also. We can get target sequence DNA, already we talk about. We can get information from metagenomics. And for RNA, especially for RNA, we study gene expression or microRNA expression. We can use small RNA profiling and gene expression, and also epigenetics like chromosome immunoprecipitation sequencing as well, and ribosome profiling. All this kind of data, we can get it from next generation sequencing. And already we talk about the database and their importance, but there are primary database and secondary database. Primary databases, those are the sequence that they are getting from the lab, from the experiments. And this is just like Urine nucleotide archive and gene bank and data bank, DNA data bank of Japan. All these are primary, where they deposit sequence in daily basis. And the good thing, all of these are now actually, you can access them from a single database, which is NCBI databases, okay? And secondary databases, those are the information that you are getting from the analysis like DB SNPs. We'll find information about the SNP, DB VAR, you'll find variants, and OMIM, you'll find all information about diseases and genes that relate to, and also about the pathways and everything. So this about pipelines, the next generation sequencing, actually there is pipelines, and pipelines are similar, but when we go down, to applications, they will be different, but we will begin from a FASTA-Q file. All next generation sequencing, they will give you FASTA-Q files, and in this, the FASTA-Q files, then you need to do what we call it a quality control. Quality control, you want to see, how you need to see if these reads are good or bad. And after that, you can do some kind of cleaning. Most applications depend on alignments. And the alignments, there are two kinds of alignments. They are reference genome guided alignment and de novo alignment. If an organism has a reference genome, then we will use a reference guided assembly or mapping. If they don't have a reference genome, they will use de novo assembly, and there are many software for that. So once you are doing that, then we are getting what we call SAM or BAM file. These the files that include the information alignment. And after that, they have different pipelines. One for all genome variant calling, if we need to know the SNPs and mutation or insertion or deletion, then we can go, we apply these pipelines, RNA-seq for gene to quantify the reads and to make what we call it differential analysis to compare between and to see the gene expression and compare between different cases. In case of ChIP-seq, here we are measuring the seq peak, and the idea is to, especially if you want to identify the binding site of transcription factors and so on. So these pipelines, and each one, by the way, is take a lot of pipeline coding and so on. So this already, I have already explained this. When we mapping actually is very important. So like immediately when we get read or long sequence, we need to map them to a reference genome, or we can use the de novo to at least to get map or file. So about the common user data format, and by the way, this is very important to understand, because that will help us to analyze. The first one is FASTA format, and FASTA format is very simple. FASTA format, it has only two parts. The first part begin with greater than and description, and we can include also unique number as accession number or and the sequence. And the sequence in case of nucleotides, and by nucleotides we mean either DNA or RNA, it should be one of the 4 alphabet, okay? Adenine, cytosine, guanine, and thymine. This is case of DNA. And in case of RNA as well, because that also will be converted into complementary DNA. RNA will be converted into complementary DNA. So in case of protein, there will be a amino acids very simple, they will be here. So they are very important, and we are using them in the analysis, but also you want to know the second format is gene bank, and gene bank include more information, include header, features, and sequence. Headers include, like, any metadata about the experiment, about the location of, and just additional information that you may need, about publication and so on. Feature, this is important section, because if you have DNA, it will include some feature like the gene, exon, CDC, like this coding sequence and so on. And the last section here, the sequence, you will find sequence in some gene bank format. You may not find sequence, but this gene format developed especially not to when you submit data to a database, to NCBI database or any one of the three databases, then you need to, if it is faster, if it is only sequence, then you need to submit in this format. FASTA-Q files is generated from next generation sequencing and third generation sequencing. And it's very important because it includes the quality of each base call. Okay, so this is really very important, and this is the raw data. You don't need to handle this. The only thing is just to take the quality and to use it in the alignment. But the format consists of 4 parts. The first part will be description. The second part will be the sequence, and then plus sign and any description. And the fourth line will be the Fred quality score that show you the quality of each of these bases. And a single file may consist of millions and millions. SAM and BAM, this includes the alignment. So when you align your read or long reads to reference genome, then you will get SAM, or you get SAM file. And SAM file has 11 mandatory fields that includes the information of the alignment. So each read and the alignment information for it, okay? And include the highlighted here are the most important field. And we use this actually in bioinformatics to get some information like flag and position and cigar string. All those are, if you are, in the future want to go more in this, because you can get really information in this just, and it will show you how it looks like the columns and so on, okay? And for protein structures, we have, we have questions. We're running out of time or what?
Yes, not a question. It's just a let that we have 10 minutes more for presentation before we go for Q&As. Thank you.
Okay. So I'll try to write down. Sorry. So so this the other actually data format is the protein structures data format, and they include the coordinates, and we get this from X-ray diffraction. And the good thing there are 2 protein structures. One is BDB and one BDBX. This one is not supported anymore, but it, as you see, it includes the atoms and the coordinates. And also the second one is BDBX. This right now is supported, and this is very flexible, and both of them actually can be used now. And they are used to visualize the structures of proteins by using some software. And the variant call format also is very important, because this, when you use variant calling or variant discovery, then you need to store this variation in this file format, because this includes the if there is the mutation as general, SNPs, single nucleotide polymorphism, or insertion or deletion, they will be stored in PDF. You can visualize it. You can make more study on it as well. So now we'll come to the sequence alignment, and sequence alignment, actually, they are very important and one of the first applications used. And we can divide them into two: pairwise sequence alignment and multiple sequence alignment. And pairwise alignment, we can divide them into global and local. Okay, and in case of global, that we need to compare two sequences, and they must be can be extended to be in the same length. But for local alignment, we focus on a specific similar group, and we're using this in homology. Multiple sequence alignment, where there are multiple sequences and we need to align them, all of them will align them simultaneously. So we will get this sequence, and each one they have their own application. So for that, we need some kind of scoring mechanism to score this kind of alignment. One of these, we are using identity score, and we use also distance, like Hamming distance. We use substitution metrics, and substitution metrics are one of the most important metrics that they are using in homology study. And in substitution metrics, we have two families. We have BAM and we have Blossom. And BAM, if we need to compare between very closely sequence, then we can use BAM, and for distance or conserved regions in sequence, then we can use a Blossom. And BAM, what you see now, the figure now, actually, this is the, we call it log odd ratio, and those are generating from the probability of substitution probability, and after that, so that means each one here is score how an amino acid will be replaced by another amino acid, and the score used in sequence alignment. So for the algorithm, in both cases, if in global sequence alignment or local alignment, we are using what we call it dynamic programming, and there are algorithms that do that, and they give you exactly the alignment using the best alignment. And for multiple sequence alignment, also there are some important application for them. Some of them position is called specific metrics. We are using it all the time. We study like homology or kind of homology, conserved region motif, and the application we are using like colossal W, muscles, and map T coffee. All those they are available, and multiple sequence alignments also we can what we call a sequence logo, and sequence logo shows you the conservative regions is the multiple alignment. And what you see here, you see each of these represented by the frequency of the aligned bilab of these, so it will give you idea about the conserved region. And one of the applications of a multiple sequence alignment is phylogenetic tree, and this is one of the most important things because it will study. And from the step they are as follows. First, you need to use multiple sequence alignment, and then the distance, generate the distance matrix, and then you build phylogenetic tree and there are many algorithms, and this is useful to show you the relationship between different, you can use this like genes or genome, but the idea it will show you the relatedness between these genes, and they can give you a visual, you can visualize this, and it will show you how these species can be related. And plus, one of applications of plus and basic local alignment search tool, we are using this for searching a sequence, especially in case of if we need a similar sequence or we need to primer or anything like that, we are using plus. So in plus there are different programs. Some of them like nucleotide plus and P plus and T plus, and each one of these it has its own use. But generally, you can first you have to know what kind of sequence. Do you have nucleotides or protein? Then you can use the sequence you have. We call it query sequence, And the important things of this, you have to choose the target database, and there are different target database that you're going to use to to search your query against this database, and there are some setting, and there's the end they will give you a result. As the result, they will show you how they will show you the most hits that found in the database, and from here you can learn that if the like homologous sequence or similar sequences, you will know the sequence, and there are some like value that show you how you can be confident in your research and identity also. And there are some parameters, important parameters that will show you the importance of your hits. And for primer designing, also we design primer for PCR for many purposes. And so what we do, also you can use Primer Plus, and you can use other software as well. And the idea is, just to find your sequence, and you determine your target sequence. But when you use that program, you need to focus on a specific thing, like the length of your primers and melting temperature, and the annealing temperature. Also, you can the annealing temperature, and then use that program. And all programs, they provide you with the default values, but also you can do some kind of adjustment. And designing program depend on what the purpose of that program is. It for PCR or other? This, if you are using Primer 3 Plus, also you can use command line as well. And this is show you how to create an input file for it and how to get an output file of it. And by the way, there are some important bioinformatics tools that I want to finish with this. You can use Python. You can use, if you are using Python, you can use PyPython, very good package for this. Bioconductors, if you are our user. You can use genome browsers and IGF, integrated genome viewer, to visualize and to explore the genome. And you can NC-BY e-utility, e-direct to search database SRA toolkits if you want to download FASTA-Q file and BAM file from NC-BY SRA databases for VDM, and BIMOL if you want to visualize protein structures, MEGA if you want to build value genetic tree, and FASTA-Q file to check the quality of FASTA-Q file. And if you want to align map reads to reference genome, then you can use one of these, BW or PAUTI or Bowtie 2 or Star, and also there are other. In case of variant calling, you can use GAC, genome analysis toolkit 4 or SAMTOL or FreeBIOS. And de novo analysis, you can use SPEDIS, APIs, or other software. For targeted gene metagenomics, you can use CHIME2. Now we have CHIME2. And for shortcut, we can use what we call Kraken or Kaidio or centrifuge. And each one of these, in fact, you can create your own pipelines, and they include a lot of details. And thank you so much. Hopefully I didn't take more time.
Thank you very much, Dr. Ismail. It's been really a very good presentation, and I like the fact that you made it to understandable to even to the novice in this area. So it's very, very clear and good presentation. We are going to have a Q&A session. So for those who have some questions or some clarifications, please do not hesitate to raise your hands, or you can just post your question in the Q&A room so that my colleagues can help us to give you the floor. So in the meantime, I can lead one. It's not a question, so to say. Someone, Birai Tafa, requesting that the presentations and the recorded video can be accessible. I just want to let you know that we, in the Academy team, we have a HoliCare Academy platform, which we have Aria shared the link for registration. for Tafa and the team that is in this webinar, you can just go ahead and register to the Academy platform. Then you will automatically be directed to the link for today's webinar, and the previous webinar resources are also there. Thank you.
Okay, Jean-Claude, if it's okay, maybe I can start with the first question.
Go ahead, please.
Comment. Yes, thank you very much, Hamid, for the nice presentation. And it's really very interesting, although I know that it might require a little bit of more explanation, especially to this some important bioinformatic tools that you just went through in the very final slides of the presentation. So I hope we would have the chance then, if not today, but later, to have a little bit of more elaboration into these very important tools. But what I don't want to miss today is, and it is related to one very important subject, which is this sequence alignment and sequence alignment tools. And you already mentioned that there are several different ways, like the cluster way, and then the other one, which is called muscle and coffee and so forth. So maybe if you can just elaborate a little bit. I excuse Jean-Claude for that. Maybe one more minute to tell us a little bit more on the differences between these methods and which one you personally may recommend, or what are the advantages, disadvantages, something like that. Thanks.
This actually is a very good question, and this for multiple sequence alignment, and cluster W may be the first one and the most one used, and now we have cluster W and also cluster WX and coffee, and each of them actually they are using different algorithm, but this is this is almost the same. All of them actually they are using the cluster W, one of me personally, one of the most ones that we use in multiple sequence alignment in biology history. But other also they prefer coffee. Other also prefer also other sequence alignment, but the difference only in algorithm, and I believe there are some papers did some kind of comparisons, especially in the accuracy of their performance. Their papers actually tell you about which one and which ones I can use and in which situation. But for me personally, I prefer cluster W.
Okay, thanks.
Maybe I can have just one novice question, Ismail, as well. Is you know, this type of webinars are somehow interconnected. I think I agree with Mohammed on this. From the previous ones and from this one and the next ones that are in the pipeline. So how my question is, how is this application of bioinformatics connected with the lamp design? Because maybe Mohammed will come on this aspect later on, but I want to know how bioinformatics application is connected with the lamp design. Thank you.
Okay, so lab design is just like for primary specifically, is just like any primer designing. So we are using the same primer design algorithm, but because especially in LAM or loop mediated isothermal amplifications, here maybe we are using not only 2 primers, forward and reverse, but we are using like maybe 6 as far as I know. And but you have to select a specific region, the regions that you need to amplify, and also you have to consider some factors that you are not using them in PCR, like especially like melting temperature and stuff like that, because this and I think there are software that they can help you to design this kind of some kind of primer for this kind of amplification, because they are different from the classic, PCR system, right? So yeah. So first, maybe you need to see your target sequence that we need to amplify, and then you need to design the 6 primer. And in these 6 primer, they must be with specific characteristics to be suitable for this kind of amplification. And there are some software that they can adjust, they can do, they can help you to do this, like to adjust on different, they are using different algorithm, and at the end they will give you the right primers that they can be used for that specific template.
Yeah, thank you. So we have one question from the Q&A Loom. It's from Zulf. He's asking, in bioinformatics, how much on new algorithm development and how much on use of bioinformatics for applications?
Yeah, actually, there's a lot of the algorithm, because bioinformatics itself, especially the computer scientists, they are busy developing this kind of algorithm, and there are main algorithms that will depend on them, like PLAS, for example, basic sequence search tools, and the Align, and the sequence genome assembly and variant calling. There are a lot of algorithms actually that are developing, and still there is active research to improve this algorithm. So it is like, it still is a continuous effort, and actually there are many, and now we begin actually to add machine learning and deep learning to bioinformatics as well. So now we are implementing, because we have a lot of genomic data, so now we are adding machine learning, artificial intelligence, all this stuff, we add them to bioinformatics. So that is still, we increasing, we are using like natural language processing, for example, we are using machine learning algorithm, all kind of machine learning algorithm in bioinformatics as well. And even in this kind of tools, the tools that we are using now, also they are using machine learning algorithm, and this that means the algorithm itself, this will be continuous. We're not going to stop, and still this is the beginning for this answer your question.
I guess so. He just posted a follow-up question about research now, saying that how much research focus now on algorithm development and how much on use, in these bioinformatics applications.
Yeah, actually, this is a very good question, because now computer science, they left the computers and now they are focusing in bioinformatics. That means that to show you how significant bioinformatics is. Okay, that means we have soldiers of, just they are going to focusing on algorithm development and so on, because this is now we are using this actually in diagnosis. Now we're going to use it actually in different things. We hear about the gene therapy, and use about different applications, and now vaccinations. They are using RNA vaccination. So they are new technology. So these attract, even computer science, that, as I said, actually they left their field, and now they are focusing in bioinformatics. That means it still are always this is the very active field of research, and attract computer scientists to be bioinformatician.
Yeah. Time, by the way, is not on our side, but we can see now more questions. One Abu Jaffer has a question like, what are some of the misconceptions about bioinformatics according to your expertise?
Misconception is actually there is the only thing to know exactly what you are doing, okay? Because is that depend on what kind of scientific questions that you want to answer, okay? Because we don't, we are focusing on DNA, RNA, and protein. If you need to study gene expression, you don't need to extract the DNA, right? Because here you have to extract RNA, okay? So this, I saw actually some student, they are extracting actually DNA, they want to know the gene expression. How come they don't know that? No, in this case, you need to extract RNA. So the conception, actually, the misconception usually it's come from the misunderstandings of what you are doing with your bioinformatics data, okay? And so if you are in DNA, what you want to do with the DNA? Maybe genome simply, maybe variant calling, you need to discover a new variant. Maybe you want to target a gene. You may need to identify a specific gene or a specific microbe. If you are using RNA, then here you'll focus in gene expressions, and also you can focus also in variant discovery. If you are using protein, also at the same time, also it will be like different thing. So this the misconception actually come from misunderstanding to bioinformatics itself. Okay, but so this is a missing, but
Great. Thank you very much. I seem not to see other questions related to the contents, but there is one question maybe which will lead us to Mohammed as we go towards the conclusion of this webinar. Some participants are asking when or how are they going to know about the next webinar and what is going to be the focus of the next webinar. So, Mohammed, if you can take that, and then as we close this webinar. Thank you.
And this very interesting bioinformatics webinar, it really acts very good for us. So we went this way into the introduction into the bioinformatics because it's always coming back as feedback from the workshop that the project did last year. There is a lot of interest from the participants towards bioinformatics, a little bit more information here, but also on assay design. And this will lead me now to the next possible webinars, which are going to deal a little bit with assay design of the isothermal amplification method LAMP, which necessitates a lot of primers and so forth, as you mentioned, but also a little bit more knowledge on sequences and how to obtain them, how to study them, and how to do the sequence alignment and so forth. Because the core for the design of any assay is not just to select the primers and where they sit in the sequence and so forth, but whether that piece of sequence which you used is fulfilling you the task for this assay. If it is specific enough, if it is it gives you the information that you need your assay to deliver later on. And this requires a little bit of information here, of course. Today, Hamid has done really a lot into this area. And for the next webinars, we're also going to take it from here and to extend a little bit on the very specific knowledge that's required in this part. And then maybe the technical part on how to do the design itself. And of course, it's also worthy to mention that the design for such assays today is really more or less automatic. There are a lot of bioinformatic tools which are helping you software to select these particular scores that you would require your selected primers to fulfill, for example, that your assay then later on is going to be fulfilling also the needs that you would like an assay to have, like the high good sensitivity, specificity, and so forth. So this is now today, let's say, the introduction part to the next one, which is going to take this further into primer design, and most likely, depending also on the feedback from the participants, we would check if we are going really also to focus into other areas or to speak more in details in specific areas in the bioinformatics that Hamid has today given the introduction into it. I hope this is good information for everybody who's interested to join us for the next times as well. Before forwarding back to you, Jean-Claude, let me maybe also once again to thank Hamid for being today really here with us. This is really great. Thank you very much once again. And also to the audience.
Thank you so much.
And I give you now back, Jen.
Thank you so much.
Thank you very much, Mohammed. Actually, this is almost close to the end of our webinar, but I can see Aman in the room. Aman, do you have anything to say before we conclude? Aman is our project coordinator, so I wanted just to know if he has something to say.
Can you hear me?
Yes, we do, Aman. Please go ahead.
Oh, yes. So this is just to reiterate what Mohammed was saying, really to thank you for a very, very nice seminar. And again, on behalf of the HoliCare Consortium, my heartfelt thank you for this very nice webinar. Hope that our way will cross in the future and to follow up. Thank you. Thank you.
Thank you so much.
Thank you, Aman. So, taking on that, I would like to thank you also, Ismail, for your time,
Thank you so much.
your dedication, and we look forward to receiving the presentation materials so that we can share among our academy platform. So this is officially to close the webinar for today. Thank you and have a nice day, everyone.
You too. Thank you so much. Bye.
Bye.